Hello biology students, and welcome to genetics video 4. So from this video on, for the rest of the unit, we're going to be talking about what's called non-Mendelian genetics. And basically this is going to be genetics that was not discovered by Gregor Mendel and does not follow his basic rules. The types of non-Mendelian genetics that we're going to be addressing in this video include incomplete dominance, codominance, and multiple alleles. So let's get started. Okay. So to kind of start out, it turns out what Mendel, discovered and taught us are absolutely valid for all sexually reproducing species. So, what he discovered is still the basics of genetics today. However, it turns out his really simple rule that if you have a dominant allele, you automatically have a dominant phenotype, and if you only have recessive alleles, you automatically have a recessive phenotype, don't always hold out entirely. So we're going to be looking at a couple of examples in this video that shows how we don't always get this simple dominant equal dominant and recessive equals recessive. So the first type of non-Mendelian genetics that we're going to look at is called incomplete dominance. So before we talk about incomplete dominance, we should probably define complete dominance. So complete dominance is what we've been talking about all along. Complete dominance is where if you have a dominant allele, you have the dominant phenotype. So in other words, the homozygous dominant and the heterozygous have the same phenotype. So maybe, if capital B is black fur and a little b is white fur, capital B, the homozygous dominant individual, and capital B, lowercase b, the heterozygote, would have the exact same phenotype. They would both be black. Incomplete dominance, however, is a type of dominance where instead of having the homozygous dominant and the heterozygote having the same phenotype, it's where the heterozygote has an intermediate phenotype. So basically, the heterozygote is a blend between the dominant and the recessive phenotype. And, this name makes sense because, we're not showing complete dominance. We're kind of almost getting there. If you have one dominant allele, you're almost to the dominant phenotype, but instead you kind of only get halfway there. So in this example, capital B would be black, little b would be white, and capital B, little b would be a blend, so it would be gray. Okay, so we look at an example. So it turns out there's a flower called a snapdragon, and if it has two dominant alleles for its color gene, it's red. If it has two recessive alleles, it's white. But if it's a heterozygous plant, it's pink. Okay, and this basically happens because if you have the dominant allele, that means you're making the red protein. And in this case, the heterozygous is only making about half the amount of red protein, so they have half the amount of red pigment, and they end up kind of a pink color. Okay, so in incomplete dominance, you always see a blending or a hybrid, halfway in between the two phenotypes. So in this case, if I took a red plant and a white plant, even though red is dominant, if I cross them, I would get all pink offspring because all the pink offspring would have one capital R and one lowercase r. And then if I crossed two F1s, two heterozygotes, I would end up with one red, two pink, and one white. So another example is hair texture in humans. So if you have two dominant alleles, you have curly hair, and the dominant allele here is being represented by C1 instead of a capital C. If you have two recessive alleles, you have straight hair, which is being represented by C2. But if you're heterozygous and you have one of each, you have wavy hair. Okay, so these are both examples of incomplete dominance because it's showing an intermediate phenotype. So the next example is codominance, and this looks really similar to incomplete dominance, but they're not the same thing. Okay, so codominance is when both, if you have the heterozygote, like here, both alleles are expressed. So it's not a blend. Instead, you show both alleles. So neither is dominant or recessive. They're both present. Okay. So for example, when we were looking at the flowers, capital R will give you a red flower, little r give you a white flower, and the capital R, little r give you a pink flower. Well, in this case, with codominance, you wouldn't get a pink flower. Instead, you would get like a red and white striped flower. Both the red and the white are present. They're not blended in codominance. So kind of a popular example of this is the rowan colored coat that can be found in cows and horses. So you can have, 2 capital alleles would give you a red horse, or like a mahogany kind of colored horse. 2 lowercase alleles will give you a white. But if you have a heterozygote, you have a horse that has red and white hairs together present. So they look kind of pink, but it's because they have both red and white present. Okay. And then finally, the last thing we're going to talk about in this video is that some genes actually have more than 2 alleles. So we've been talking about this simple situation where you either have a capital letter or you have a lowercase letter. But in reality, think about things like eye coloring. You don't just have blue or brown eyes. There's lots of colors in between. So you could have more than 2 alleles for a gene. So kind of the quintessential, really famous example of this is blood typing. So there are 3 alleles you could have for blood type. You could have the allele capital I superscript A. Okay. You could have the allele capital I superscript B. And then you could have a little I allele. There's 3 different alleles for blood typing. And it turns out these 3 alleles can make 4 phenotypes. So you might know there are 4 blood types. There's A, B, AB, and O. And kind of the relationships that exist between these alleles, I superscript A and I superscript B are codominant alleles. Which is why, if your genotype was IA, IB, you would have AB blood. And then little I is recessive to both IA and IB. Okay, so we can think of some examples. If I was, if my genotype was IA little I would have A-type blood because the little I is recessive to the A. And if I had little I, that's how I would have O-type blood. So let's look at a couple more examples and information about blood typing. Okay, so it turns out if you are type A blood, what this means is that your genotype is either IA, or it's IA little I. And what this means is your red blood cells have a protein on them called the A antigen, okay? And then in your blood, there are antibodies that will attack the B antigen. If you have B-type blood, you have the B antigen on your blood cells, and you have antibodies in your blood that will attack the A antigen. So you might know that, if you're getting a blood transfusion, if you're getting blood added to your body because, you had an operation or you were in an accident, you have to be very careful what type blood is given to you because, if you're type A blood and you're given type B blood, the antibodies in your body will attack the type B blood. And the reason for this is these antibodies are basically here to recognize foreign things. Generally, you don't want to have things that are foreign in your body, so your body has this defense mechanism up to attack them. Okay? If you're type AB blood, you have A and B on your blood cells, and you have no antibodies in your plasma, which means you can receive any type of blood because you don't have any antibodies to attack anything else. And then finally, type O blood, you have nothing on your blood cells, but you have antibodies that will attack A and attack B. So type O blood can't receive blood from AB or AB because their antibodies will attack them. However, type O blood, when you donate blood, you only donate the cells. So type O blood is the universal donor because they can give blood to anyone. Because I can put this blood cell into any of the other types and it wouldn't get attacked because there are no antigens on the surface. So to kind of summarize, if you're blood type A, you have A proteins on your blood cells, and you have antibodies to the B protein type. So you can only receive blood from type A or O. Okay, but you can donate to type A or AB. And you can kind of go down this chart, and you can see that O-type blood can donate to everyone because no one's going to attack cells that have no antigens. And AB can receive blood from everybody because it doesn't have any antibodies to attack anything. So that's it for video 4, and I'll see y'all in video 5.